Hi guys, this is part 2 of the biotechnology lecture. One final technology that I want to share with you guys is something that allows us to edit the DNA, and it's a system called CRISPR Cas9. And before I describe the details, I just want to give you a general idea of what this is about. So you have already learned about restriction endonucleases. Restriction endonucleases, again, are these collection of enzymes that will recognize a very specific sequence, and whenever they see that sequence, they will cut the DNA and on both strands. And there's a bunch of different restriction endonucleases that will recognize a different sequence, but they are limited to that sequence. EcoR1 can only cut at this sequence. You cannot change what that sequence is for EcoR1. Cas9 is another type of endonuclease, so it can cut DNA, but where it cuts DNA is flexible. What actually tells Cas9 where to go is a piece of RNA. Now, in the laboratory, scientists call it a single guide RNA, and I'll explain that in a minute. But this piece of RNA, depending on what sequence you make it, it can guide Cas9 to almost anywhere inside of the genome or in the DNA. And so you can basically engineer where this enzyme is going to go cut. So that makes it a much more flexible enzyme than the restriction endonucleases. The CRISPR-Cas9 system was originally identified from bacteria. It's a type of adaptive immunity, actually, if you will. What this system allows bacteria to do is it enables them to respond to and eliminate foreign invading genetic material. So for example, there are viruses that will go and inject their DNA into bacteria. And the idea is they want the bacteria cells to start making more virus. But to defend themselves, bacteria can unleash CRISPR, or the Cas9, that enzyme, to go and cut it up so that it doesn't do this. So let me describe in brief what this system looks like in bacteria. There are 3 core elements. The first component that I'll mention is the CRISPR locus. And so if you look up here, this is a piece of or stretch of DNA that you would find in the bacterial genome that contain this system. And this area of the DNA, this CRISPR loci, what it contains is DNA sequence that came or remnants or pieces from former invading foreign DNA. So what happens is when foreign DNA comes in, the bacterial cell will chop it up into small parts and will save some of it in its own genome. And that's what you see in the green and blue. In between these remnants of old invaders are repetitive sequence, and they call it CRISPR RNA because once it's transcribed into RNA, they're going to call it CRISPR RNA. But this DNA is repeated over and over in between that remnant DNA of an old invader. The second part is called tracer RNA. So this is a piece of DNA that after transcription will make a piece of RNA that they call tracer RNA. When this RNA is made, it's going to be partially complementary to the RNA that is made from these little repetitive segments right here. And I'll show you that in a picture on the next slide in a second. But this is partially complementary to this. They're going to interact. Okay, you'll see. The third part is this gene right here. It's the Cas9 gene, and it will produce the Cas9 endonuclease, the enzyme that will cut DNA. So this outlines a series of events that would happen in bacteria should for example, a virus inject its foreign DNA. So here's the foreign DNA that comes in. What would then happen next is in bacteria, this invading DNA would get chopped up into small bits, small pieces, like 20 base pairs long. And then a piece of the foreign DNA will get inserted into that CRISPR locus I told you about. and it would be inserted between a couple of repeats, those little CRISPR repeats that I mentioned before. Again, this is a way for the bacteria cell to remember who invaded it in the past, so that if the bacteria cell ever got invaded again, so let's pretend this is the same DNA and it's invading again. If that happens, then now the CRISPR locus is going to be transcribed, it's going to be turned on, and you'll get a piece of RNA like you see down here. It says primary transcript. This is a piece of RNA that was made now from the CRISPR locus in the DNA. The tracer RNA will also be transcribed, and so you see that here. And if you remember, part of the tracer RNA is complementary to the CRISPR RNA. So they are going to interact, they're going to hydrogen bond. And so you see that here. The tracer RNA will hydrogen bond with any CRISPR RNA, or those repeats. Then this large CRISPR RNA is further processed so that you effectively get a piece of the CRISPR repeat RNA with the unique remnant of the foreign DNA, and you have a tracer RNA that is binding to it because it's complementary to that CRISPR RNA. So they're only showing you the one piece that is relevant to the foreign invader that returned. The tracer RNA plus the CRISPR RNA with the piece of foreign DNA, that whole thing together, that's what we would call a guide RNA, or the single guide RNA. This sequence is what's going to then complex with Cas9 and will guide Cas9 to a complementary sequence to this unique piece of RNA here. Whatever it finds it's complementary, it's going to then encourage Cas9 to cut the sequence it found and bound to. It's going to cut it and effectively inactivate it. That's the goal in bacteria cells. You want to inactivate this foreign DNA. This image is just a zoomed in depiction of what would be going on. So in yellow is the Cas9 endonuclease, the enzyme. In light gray here is the DNA sequence that it was guided to cut. And you can see that one strand of this DNA is complementary. So this one strand here is complementary to the guide RNA. So this guide RNA is supposed to represent the CRISPR RNA plus the tracer RNA plus the unique RNA sequence from the previous invasion. So since that unique sequence is complementary, Cas9 was guided there, and Cas9 would then be encouraged to cut the DNA that it was complementary to. And it will cut both strands like you see here. And in essence, like we said, it's inactivating that piece of DNA. On this slide, I just wanted to show you that although in bacteria, in the normal system, you needed three things, right? We needed the CRISPR RNA that had the unique sequence attached to it, and it had the tracer RNA that was partially complementary to it, so they would complex together like this. And then, of course, you had the Cas9 endonuclease. So you needed all three of those things. When we use Cas9 as a tool, or CRISPR Cas9 system as a tool in the lab, what scientists do, they'll use plasmids like this. So let's say we wanted to use this system to cut a sequence in a human cell. Human cells don't have Cas9, they don't have guide RNA or anything like that. So we have to put the whole system into our cells, into a human cell. So companies have created plasmids like this one that have everything you need. So for example, here's the gene for Cas9, and it's under the control of a promoter. So if you put this plasmid into a cell, it should turn on Cas9 and make the endonuclease. There is an origin of replication, there's an ampicillin resistance gene for selection. And then up here you have a promoter, and what the promoter is controlling up here is two things. So one it says target sequence, and it just has a bunch of ends. This is the piece that the scientist could engineer. This sequence in question is what's going to be here. We as scientists could make this any sequence we want. We can control, in other words, where Cas9 endonuclease is going to be guided to, because this is the part that's going to be used to help Cas9 find something complementary in the DNA so that it can cut it. So scientists can put whatever they want in that spot. And it will be parked right next to what they call the guide RNA scaffold. That's this part right here. So scientists have put these two pieces as one whole piece together, and they call it guide RNA. So that's why you see the little G, guide RNA. So this whole thing together is going to build this. So you have the Cas9 and you have the guide RNA. All you have to do as a scientist is engineer what this sequence is going to be, and that's going to go here. So once you have that all built, you can take your plasmid and stick it into the cell of interest. And so let's say you did that, right? So let's say that we put this system, we put that plasmid into a human cell, for example. So this is now happening in a human cell. And your Cas9 enzyme, the yellow blob, was guided by the guide RNA here. We engineered what the sequence is going to be. So it guided Cas9 to this piece of DNA here, and we cut it. What happens now? What happens now really depends on what is the scientist's goal. Let's take two examples. In the first example, let's say the goal of the scientist was to mutate a piece of DNA. Maybe they wanted to create a mutation. If that's the case, then what you might do is, again, create the system so it will target Cas9 to cut the area of the DNA where you want to mutate. And then what happens next is you're just going to allow the cell to say, oh my gosh, there's a broken piece of DNA. I have to fix it. Cells do not like broken DNA. They do not like double strand breaks. They want to fix it. So then if you just let the cell naturally do its repair of the break, if we're talking cells like human cells, whenever human cells have a break like this, and that's just showing this broken piece right here, so it's broken now because Cas9 cut it. One of the major DNA break repair systems that happens is called non-homologous end joining, or NHEJ for short. This type of repair will just look at the break and try to shove the two pieces back together. But in that process, you might accidentally lose a few nucleotides on the ends, so you might be missing some sequence in the middle, or it might accidentally insert a few nucleotides. Either way, by just trying to shove the two broken pieces back together, it's very likely it will create a mutation there. NHEJ is a mutagenic form of DNA repair. So just letting the cell correct the break on its own, you're more than likely to get a mutation. So that's one option. You just cut the DNA sequence using the CRISPR Cas9 sequence, then allow the cell to repair it using this mutagenic form of repair. And voila, you created a mutation. But let's say you have a different goal. Maybe you as the scientist, your goal is you want to change the sequence that's there in the DNA. You don't just want to mutate it. You want to make a specific change. Maybe it's already mutated and you want to fix it. Or maybe you want to make a more specific mutation. In that case, if you want to do more fine editing like that, you're not going to let the cell rely on this type of repair. What you would have to do is, in addition to providing the cell with the CRISPR Cas9 system on the plasmid like I showed you, you would also have to give the cell what we would call a donor piece of DNA. This donor DNA would contain DNA sequence that is homologous or similar to the sequence you cut. So the cell will think, oh look, here is a sequence that is like the same sequence I just broke, or that is broken. Let's use that sequence to copy off of to repair our broken one. It's going to think, oh, this is something we can use to copy off of. But you can trick the cell and put inside of your donor DNA that is homologous, right? You can see by the green color that means they are similar sequence. But in the middle, you can squeeze in sequence that is new, that is different than what was originally present. And that's what's in blue. So as the cell tries to repair the break by copying off of the donor, it's also going to copy this new sequence and incorporate it into its own DNA. And so, like I said, you could make this sequence maybe the correct sequence that's supposed to be there, or maybe you're inserting new sequence. Either way, you can control what happens to the DNA, the original DNA sequence. You can insert something brand new. This type of repair is called homologous recombination repair. So just to recap, in the second option, you would be cutting the DNA sequence with the CRISPR Cas9 system, right? You can guide it there using that system. Once it's cut, the cell wants to fix it. But if the cell is provided with a donor sequence that is similar in sequence to what you cut, so they're similar to each other, they're homologous to each other, the cell will say, oh, hey, look, we can copy off of this sequence. And so it will opt for homologous recombination repair instead of non-homologous end joining. So if it does that, it will copy off of the donor, or it will use the donor sequence to repair it. And in doing so, it will incorporate anything new that you sneak in there. And so you have now more, you've conducted a mutagenesis in a more specific way. And therein lies the power of the CRISPR Cas9 system. You can either make random mutations into your sequence of interest, or you can make more direct mutations or specific mutations into your DNA of interest.
And just to give you guys an idea of what is being done with CRISPR Cas9, or this system in humans, the first ever USDA approved CRISPR edited organism was a mushroom that was genetically modified to resist browning. The first clinical trial ever involving CRISPR was in a West China hospital. In 2016, the doctors removed immune cells from the blood of a person with lung cancer, and they tried to use CRISPR to disable a gene and then return the cells back. And this gene is like an off switch for immune cells that cancer cells can use. Also, there was an attempt to use CRISPR to edit cells while they were still in the body. And what they were trying to do was to prevent cervical cancers by targeting and destroying the genes of the HPV, or the human papillomavirus, that cause tumor growth. That was also being done in China. And of course, there was that big controversy that happened over a year ago now, where a Chinese scientist edited human embryos to contain a mutation in the CCR5 gene, which would render these individuals resistant or more resistant to HIV infection. So CRISPR is being used in many different ways currently, and in many different research studies. And since CRISPR came out, there's been a lot of variations on this technology, but I wanted to show you what the original technology involved.